Okay. So hello everyone. So I am Vishwambar Navle from India, and I am a PhD student working under the supervision of Dr. Koteshwar Rao. So today I am going to present my talk on the topic management and detection of toxigenic fusarium species and their mycotoxins in food grains. So let's start with the introduction. As you all of know that these fungi are the member of eukaryotic groups, and there are about 8,000 fungal species are known as a plant pathogen, and near about 300 are the human pathogens. If you see the major pathogenic and toxigenic fungi, in that one particularly Fusarium, Penicillium, Aspergillus, and Alternaria. These are the major genera which are pathogenic, act as a pathogenic as well as toxigenic, and they produce a diverse type of mycotoxin. These mycotoxins, these are nothing but these secondary metabolites produced by these fungi, and they have a diverse structure. They are thermostable, non-antigenic in nature. So if you consider like agriculturally important mycotoxins like ochratoxin produced by Penicillium and Aspergillus, aflatoxin well known produced by the Aspergillus genus, then this trichothecin, zearalenone, and fumonisin, these are the particularly produced by the Fusarium genus. And according to the Food Agriculture Organization, so more than 25% world food crops they are the contaminated with mycotoxins, and these mycotoxins they causes various acute and chronic diseases in humans and animals. So these are the diverse structure you can see here. These are the diverse structure of these mycotoxins and their particular backbone, like in case of zearalenone. uh, so this backbone is responsible for their toxic effects to the humans and animals. So here, if you see the overall contamination, so food additives like different chemicals, veterinary medicines, pathogenic microorganisms, and mycotoxins. So in overall things, if you see the more than 40% mycotoxins are the major contaminant found in food and feed system. So and these mycotoxins, if you see, so these are the different mycotoxins, and they cause different organ diseases in humans as well as animals, like organ damage. And if you see in case of the plants, they enhance the pathogenicity of that particular fungi. So this fungus and their mycotoxins, these are the most neglected part by the various funding agencies and even resources also. So there is a particular issue on the nature microbiology. They clearly reported mentioned that these fungi, so stop neglecting these fungi because they are the major affect human as well as animals health. So now we'll see the how the mycotoxin got produced and in food and feed system. So this for the mycotoxin production, there are various biological, intrinsic, physical factors are there. So these all things are they are responsible for the its production. So if you see the intrinsic factors like those fungi, they are able to produce these mycotoxins. They are like strain specificity. Some are non-mycotoxigenic, some are able to produce. They are like also host specific. Host specificity also there, like fusarium. If the major host is the maize, so it will maize and wheat. These are the major hosts. So it will these are the intrinsic factors for responsible for this mycotoxin production. If you see the chemical factors like carbon dioxide, oxygen, pesticides, different type of fungicides, so this factor also causes stress to that particular fungus. So it starts for the mycotoxin production. And as environmental factor like temperature, moisture conditions, relative humidity. So these things are the majorly responsible for the mycotoxin production. And after that, even at field level also it got produced. So once it transferred from field to the storage, like storage go downs. So at that time traveling, the time or moisture condition, there are also some physical environmental condition are different. So at that time also these fungus they are able to produce the mycotoxins, and these mycotoxins like transfer from uh, like grains to animal feed. And this animal feed also it transferred through field because there is no detoxification strategy, so it transfer in animals, and these animals like chickens, cow, and their products also consumed by humans. So finally humans and animals are the consumers of these mycotoxins. If you see the global occurrence of the risks of this mycotoxin, so Biomin in 2019 they did the survey, so they find out that the most, if you see the extreme risk and moderate risk of this mycotoxin occurrence, the Asian countries mostly they are in a extreme risk of the mycotoxins. So if you see the other like North America and these things, they are the about the moderate risk of the mycotoxin contamination. So now we'll discuss about the how what are the methods are used for the detection of these mycotoxins, and how they are the sensitive for their detection. If you see the analytical methods like TLC to mostly advanced one LCMS, MS, these are the GCMS. These are the various methods used for the detection and quantification of mycotoxins in food and feed. And if you a rapid diagnostic method, if you see like immunoaffinity based techniques are there like ELISA, LFDs, various types of biosensors are now available. And the emerging technology, proteomic based detection, also molecular techniques like a real time PCR, also normal PCR also there. So these are the different techniques used for the detection of mycotoxins. But here the regarding the like developing countries like India, here the these LCMS and these rapid diagnostic ELISA or these kits are so expensive. So PCR based detection is the like cost effective method for the early detection of mycotoxin fungi in food and feed. So considering this problem, we have developed the multiplex PCR for the simultaneous detection of mycotoxigenic fusarium species. So for this one, first of all, we designed the primers, those having the more specificity towards the target gene. So, and this individually, those are known mycotoxin producer strains we have isolate collected from the different natural collections, and they are the well known for the mycotoxin production. So, firstly, we optimize the monoplex PCR with the different genes. So, here three genes we majorly targeted: fumoninsins, mycotoxin, zearalenone mycotoxins, and trichothecenes. And these three are the major mycotoxins produced by Fusarium species. So after that, we check the specificity and the sensitivity of this multiplex PCR with spike the known spores of these fungi in the food system, and then we again isolate the genomic DNA from that, and then we check the specificity. So here with spike one, we found that around 10 raised to the power 2 spores per gram of the grains can be detected, or can we can detect the mycotoxins fungi. So, but in natural, if you see the natural naturally contaminated grains, so here also we develop, but here are around 10 raised to the power 3 spores per gram of the maize grains is able to detect whether these grains are contaminated with particular mycotoxinic fungi or not at genome level. So what this multiplex PCR is a highly specific and reliable and a sensitive approach that could be used for the simultaneous and early detection of this toxigenic fusion strain in store food matrices. So further that also, because from various sites of India, 10 different sites, we collected the maize samples, and we process for the mycotoxin detection as well as the quantification. So these maize samples finally grounded into a powder, then we have extracted with the different solvents and then passed through the immunoaffinity column. So and this final extract we analyze by the LCMS base approach. So here, if you see, these are the different sample sources from India. We have collected about 100 samples, and we quantify the amount of toxins present in that particular maize samples. So here, if you see the fumoninsins, don, nivellinol, and zearalenone, these are the four different toxins we quantify, and these all of the toxins are like they are they showing the more limit of their presence in maize grain, because this European Commission, they set a particular limit. So all these toxins, they are crossing the European Commission limits. So from that, it's what conclude that the Indian maize used in the poultry and uh, animal feeds, they are highly contaminated with the mycotoxins. So next, considering this challenge, now the management, what are the management strategies we have to use or apply for the these mycotoxins? So there are various methods we can use or the for the management purposes. Even at the pre harvesting times, or the proper harvesting strategy also helps to uh, manage this mycotoxin contamination. So appropriate harvest season, their schedules, right post harvest and storage conditions, biocontrol agent if you use, and use of fungis means limited use of fungicides, and that also helps to manage or prevent the mycotoxin production. So if you see the major physical, chemical and biological, these are the three methods used for the mycotoxin management. So in physical methods, like some traditional methods like sieving, thermal heating, used for the treatment, so that avoid the fungal growth and mycotoxin production. Like and in case of chemicals, various chemicals are used, but this both physical and chemical methods have some um, drawbacks. So because sometimes this grain quality got affected, and even this, if you use the chemical agents, so some other byproducts are formed, so that again act as a more harmful to the humans and animals health. So biological control, like use of the various organisms, plant-based molecules, and another you can say the some those bioactive molecules that also act as a uh, eco-friendly management strategy. So for this one, we are working on the biological based approach. So if you see the this fusarium species produces this diverse type of mycotoxins and contaminated the grains that affect the humans and animals. So these are the different toxic effects caused by these mycotoxins. So here we are particularly targeting the lactic acid bacteria, because you all of know that this is a lactic acid bacteria. They are generally recognized as a safe, and they have be diverse type of application against this, even against these fungus, their mycotoxins, and various bacterial pathogens also. So they produce organic acids, cyclic peptides, antioxidants, some fatty acids, some important molecule like ruterin, and they they inhibit the growth of fungus, and as well as detoxify the mycotoxins. So considering these things, so we are particularly targeting the lactic acid bacteria as a management for the mycotoxins. So what is the mechanism behind this? So there is no particular mechanism yet discovered, but many researchers, they propose the mechanism how this lactic acid bacteria, they are involved in the mycotoxin detoxification. So as you know, this lactic acid bacteria is a gram positive bacteria, so they have a cell wall component like peptidoglycan, thicoic acid. So many researchers propose that these mycotoxins, they are mostly detoxify or decontaminated by this lactic acid bacteria by their binding mechanism. So whatever mycotoxin, they interact with the cell wall component of the lactic acid bacteria, and further their internalization, that in which the intracellular proteins that helps to detoxify these mycotoxins. So for this one, we have collected around more than 100 sample, different dairy samples, curd, milk, cheese. So from that, we have isolated the 174 strains. So among this one, we check their activity against their antifungal and antibacterial activity. So among these strains, the Lactococcus lactis, Enterococcus faecium 2 strain, and one Enterococcus cloaci 1 strain. So these strains we have isolated and characterized. So these are the major strain that acting against the fungi and bacteria we found. So further we check their probiotic characterization of this Lactococcus species. So and we found that this probiotic characteristics like their survivability under low pH, gastric conditions. So we check these all the probiotic characterization, and we found that it survive at low pH, even at low pH, that is 2 to 4. Its survivability around 75 to 78%, and even in bile salt concentration 0.3% we use. In that one also we got around 75% survivability, and the pancreatin 0.5% we use. So with that one also it can survive about the 75%. So even after this CFU also we counted, and here you can see the bile salt tolerance, and this is the control one, and this is the test one. So you can see here the survivability of this strain. So after that one, even we tried at different temperatures. So this strain can resist, can grow from 30 degree to 45 degrees Celsius. So and the optimum temperature is the 35 degrees Celsius. So after that one, we also check their autoaggregation and coaggregation properties. So it shows around more than 60% autoaggregation properties, and the coaggregation with like E. coli and Bacillus. So with coaggregation with E. coli around 45, more than 45% it showed the coaggregation, and with Bacillus it showed the more than 55% coaggregation. And after that one, it is important that whether this strain having a hemolytic activity or not. So we grown this strain on the blood agar medium. So and here we can see that there is no zone of inhibition. So it means that it won't having any kind of hemolytic activity, and it is we can consider this is a probiotic bacteria. So it will not harm to the humans. So further we characterize the these molecules produced by this lactic acid, Lactococcus lactis. So in multitude of analysis, we found that this bacteria it produce around 1 to 2 kDa molecules, and these 1 to 2 kDa molecules are reported as antifungal agents, but their further characteristics is required, because of further particular their by purification and its activity we have to check. So and the GCMS profiling of this cell extract, we found that this bacteria produces different molecules, and in that one, among that one, 5-chlorobenzimidazole, 2-carboxylic acid, squalene. These two are the novel compound we reported from this Lactococcus lactis species, and their further characterization is going on. So after that, we check their antimicrobial potential. So this bacteria having a good antibacterial activity against E. coli and Bacillus subtilis, and so and it also having the like DPPH free radical scavenging activity. And further, if you see here, the antifungal activity against Fusarium verticillus, which is well known for the mycotoxin production, Candida albicans, Aspergillus flavus. So this bacteria having a good antifungal and antibacterial activity. So further, in vitro, how this bacteria helps to detoxify mycotoxin, that really we want to see. So for that one, we have designed the experiment in that one. The here in probiotic cultures we grown in the MRS media, and after that one, both cell-free supernatant and cell pellet we separate out, and the peptide solution among this cell-free supernatant, we isolate the means we purify the peptide solution that actually we use for the detoxification purpose, and the means here two cell pellet and peptide solution both we use for the detoxification of mycotoxins. So known concentration of mycotoxins we added along with the cell pellet and the peptide solution. So where we found this initially two toxins we took. One is the zearalenone and another is deoxynivalenol. So those both are produced by the Fusarium species. So in both case, we tried the different lactic acid bacteria like Faecium, Lactococcus lactis, Pneumoniaceae, Klebsiella pneumoniaceae, Lactobacillus vulgaris, lactobacillus acidophilus. So these are the different with our isolate, and some isolate we got from the culture national culture collection. So we check the which strain give the good detoxification of these mycotoxins. So here we found that our isolate is giving the more than 70% detoxification of both the toxins with their cell pellet as well as a peptide solution. So further, we want to check at field level, at in vitro with maize grains, whether this bacteria really works for detoxify these mycotoxins or not. So for this one, we design an in vitro experiment in which maize grains we sterilize and inoculated with the known spore with of Fusarium graminerum, which is well known for zearalenone production. So above maize grain then freeze lactococcus lactis freeze dried cells about 0.25 to 3% we use in this experiment, and water activity was maintained around 0.70, and this incubated for 21 days because this toxin production will start after the 7 days of incubations, but still we monitor it up to 21 days. So after the completion of incubation period, we extracted the zearalenone and quantified using HPLC. So here we can say we found that after the even these bacterial freeze-dried cells, 1.25% cells, if we use that, also start the inhibition of the detoxification of this zearalenone. Means this Fusarium gramineum, they are unable to produce these mycotoxins if we apply this freeze-dried cells of Lactococcus lactis. So, in conclusion, we developed the multiplex PCR assay that is highly specific and sensitive approach and could be used for the rapid detection of toxigenic fusion species in food matrices. So, our current finding indicates that there is a severe contamination of Fusarium mycotoxins in Indian maize, which has adverse effect on humans and animals' health. The present study we demonstrated that Lactobacillus lactis has uh, potential activity, and they act as antioxidant. They're having various activity like antioxidant activity, inhibitory activity against bacteria and fungal growth. It also inhibit the ergosterol and zearalenone biosynthesis. The freeze-dried cells of these Lactobacillus lactis showed the significant inhibition of zearalenone biosynthesis. So, but the further actual mechanism at a molecular level need to examine. So the, and our GCMS profiling of this strain, it produces the bioactive molecules like 5-chlorobenzimidazole, 2-carboxylic acid scoiling. So these are the like first time we are reporting from this strain, and we recommend this Lactobacillus lactis strain as a promising probiotic bacteria, and it can might be used as a novel biocontrol agent that will substitute the antibiotic growth promoters, which is routinely used nowadays for the for food in food and feed. So, lastly, I acknowledge my supervisor, Dr. Koteshwar Rao, for his support and guidance, our head of biochemical science divisions and director CSR INCL, and funding agency SCRP and DST, and our organizing committee. They are giving me like chance to present my work here. So, thank you all, and stay safe from COVID-19.